Dear students, today we will describe in detail the Oparin and Haldane experiment. According to their theory, life evolved in the oceans during a period when the atmosphere was reducing, containing hydrogen gas, water, ammonia, methane and carbon dioxide. What? No free oxygen was there. In the early decades of the 20th century, Alexander Oparin in 1924 and John Haldane in 1929, before Oparin's first book was translated into English, independently suggested that if the primitive atmosphere was reducing, means as opposed to oxygen rich atmosphere, and there was an appropriate supply of energy such as lightning or ultraviolet light, then a wide range of organic compounds might be synthesized. Oparin suggested that the organic compounds could have undergone a series of reactions leading to more and more complex molecules. He proposed that the molecules formed colloid aggregates or conservates in an aqueous environment. The conservates were able to absorb and assimilate organic compounds from the environment in a way reminiscent of metabolism. They would have taken part in evolutionary processes, eventually leading to the first life forms. Haldane's idea about the origin of life were very similar to Oparin's. Haldane proposed that the primordia sea served as a vast chemical laboratory powered by solar energy. The atmosphere was oxygen free and the combination of carbon dioxide, ammonia and ultraviolet radiation gave rise to a host of organic compounds. The sea became a hot dilute soup containing large populations of organic monomers and polymers. Haldane envisaged that groups of monomers and polymers acquired lipid membranes, and that further developments and eventually led to the first living cells. Haldane coined the term prebiotic soup, and this became a powerful symbol of Oparin-Haldane view of the origin of life. Now, we will discuss another experiment that shower light on the prebiotic environment to the journey of evolution. This is the Urey and Miller experiment. In 1952, Harold Urey tried to calculate the chemical constituents of the atmosphere of the early earth. He based his calculations on the widely held view that the early atmosphere was reducing one, and concluded that the main constituents were methane, ammonia, hydrogen gas and water. He suggested that his student Stanley Miller should do an experiment attempting to synthesize organic compounds in such an atmosphere. Miller carried out an experiment in 1953 in which he passed a continuous spark discharge at 60,000 volts through a flask containing the gases identified by Urey along with water. Miller found that after a week most of the ammonia and much of the methane had been consumed. The main gaseous products were carbon monoxide and nitrogen. In addition, there was an accumulation of dark material in the water. Few of the specific constituents of this could not be identified. What? It was clear that the material included a large range of organic polymers. Analysis of the aqueous solution showed that the following had also been synthesized: 25 amino acids, that is the main ones being glycine, alanine and aspartic acid, several fatty acids, hydroxy acids and amide products. The Urey-Miller experiment was immediately recognized as an important breakthrough in the study of the origin of life. It was received as confirmation that several of the key molecules of life could have been synthesized on the primitive earth in the kind of conditions envisaged by Oparin and Haldane. These molecules would then have been able to take part in prebiotic chemical processing leading to the origin of life. Since the Urey-Miller experiment, a great deal of effort has been spent investigating probiotic chemistry. It has become apparent that organizing simple molecule into assemblies capable of reproducing and evolving is a far greater task than was generally realized during the excitement that followed the experiment. In addition, the view that the early atmosphere was highly reducing one was challenged towards the end of the 20th century and is no longer the consensus view. Although the significance of specific details of the Miller-Urey for the origin of life may now be in question. It began the new scientific discipline of prebiotic chemistry and has been enormously influential in the development of ideas about the origin of life. In similar experiments, others observed formation of sugars, nitrogen bases, pigments and fats. Analysis of meteorite content also revealed similar compounds, indicating that similar processes are occurring elsewhere in space. With this limited evidence, the first part of the conjecture story, that is chemical evolution, was more or less accepted. We have no idea about how the first self-replicating metabolic capsules of life arose. These capsules reproduce their molecules perhaps. The first cellular form of life did not possibly originate till about 2000 million years ago. These were probably single cells. All life forms were in water environment only. This version of a biogenesis, that is, the first form of life, arose slowly through evolutionary process from non-living molecule, is accepted by majority. However, once formed, how the first cellular form of life could have evolved into the complex biodiversity of today is the fascinating story that will be discussed in coming episodes. So, my dear students, today we have discussed in detail the Oparin and Haldane experiments that told that the life evolved in the oceans during a period when the temperature was reducing. The similar experiment was also carried out by Urey and Miller, which shows that the atmosphere during the origin of life was also reducing one. The molecule formed colloid aggregates or conservates in an aqueous environment. The conservates were able to absorb and assimilate organic compounds. In the next episode, we will discuss about the evolution of human beings in detail. That is, how do we have evolved from the different time periods. Thank you.